Oh, wow. Apparently, if you start recording previously and don't turn it off, it starts up automatically. Which is good. This will be a good one to have recorded, I think. It's always so quiet. I guess, everyone being muted does that.
We need some icebreakers.
Oh, what's the weather for everybody today?
Gorgeous here in East Lansing.
Yeah, it's nice.
Sunny, blue skies, clear.
I kind of assumed like half the people weren't in Michigan, so.
Yeah, probably about half. I wonder where everyone is.
Out enjoying the Michigan weather, most likely.
I doubt that.
Hello.
Howdy.
Hi.
Yeah, we're beginning to wonder where everyone is. Sorry, go ahead. Victoria, were you saying something?
Oh, sorry. I was just saying, is a paperclip from somewhere, like a movie? It looks
Oh my gosh. It's Clippy.
I take it you are a Mac user.
Oh. This is I think this is very much a generational thing.
Yeah.
I don't even know how to explain the idea of the Microsoft Office assistant.
Yeah, so Microsoft Office in the late 90s through early 2000s, if you were going to use any Microsoft Office product, Clippy was there to tell you what to do and to try and help in the least useful way possible, no matter what you're trying to do.
So like Cortana 0.1.
Yeah, although I think there was a version before Clippy that was even worse. That was just there briefly.
Like, you know, the like little squiggly lines for like spelling or grammar would like pop up and be like, it looks like maybe you want to spell this way instead.
Oh, he actually pops up?
Yes.
Up next to things, yeah. With like little animation.
Every time you misspell anything or do anything that it looked like you were trying to get something done.
Basically, did you ever play Legend of Zelda Ocarina of Time? If so, do you remember Navi? It's exactly like that.
Just like Navi, yeah.
Except with occasionally a higher pitched chime.
We have to start giving points for people for arriving on time or something, right? Because, points, that helps everything.
You make a sticker chart.
Stickers, yes.
I do apologize. It took me a moment to find the correct Zoom link for today, and then Zoom made me install things. So
wonder. I realize that when I look at all of your faces, that is on a monitor above where my camera is. So I apologize if I'm looking up a lot. But when I actually start talking today, all the slides will be up there too. So I will very much be focused on that. But
It's okay. You can stare at your monitor. I will stare at the students.
excellent. So the goal for today's workshop is going to be to talk about some really basic genetics and a lot of the ideas on the biology side that we're trying to recreate in a digital medium. Although, you know, we are abstracting many of these ideas and we are shifting many of them to be much more focused on application in many cases and that sort of thing. But it's important to understand the underlying biology if we're going to know how we're abstracting it and where we're deviating from it, right? And so Mike and I are going to be going back and forth quite a bit with this. I've spent lots of time, actually Mike has spent probably even more time than me now, but talking to computer scientists about biological concepts. And I will often do it from the perspective of a computer scientist. So I'm someone who, until I entered my PhD program, I had not taken a biology class since middle school, right? Like, because I took high school biology in middle school and then didn't take anything through college. So it was when I jumped into my graduate level neuroscience course was the first biology course I had in any kind of real way. So I very much identify as a computer scientist who came to biology much later in my own academic career and feel very comfortable explaining concepts in terms along the way that I first understood them and that made sense to me. Mike, of course, comes from the opposite direction, where his background is all very much on evolutionary biology and you know, biology broadly speaking. I don't know, Mike, I guess I should let you say a few words about yourself.
Sure. So all of my formal training is in biology. My knowledge of computer science, I learned to program Logo when I was a small child in the various weird competitions we had in my area. Most of my programming is honestly done in R because I learned that for useful statistics. But for the past several years, I've taught a class of biology for computer scientists. And I've also been involved in the artificial life community, so I'm fairly comfortable explaining biological concepts to people with an engineering background.
So one of the important things is this should be a very interactive session. So if anything we're saying isn't making sense or whatever, please interrupt. We are more than happy to have anyone jump in at any point with other questions or anything.
In particular, if we use words that you are not familiar with. Charles likes to point out that the typical student in an introductory biology course in college learns more new words than the typical student in an introductory foreign language course. So if we use a term you're not familiar with, flag it and we will explain.
And for those of you who do have a little bit of a biology background, I'll apologize in advance if this is too basic for you, right? Because we're really going to be covering a lot of the really simple stuff in genetics. The session is being recorded, and I realized that last time I recorded and never put it online, so I should put both the last one and this one online. Yeah. Any questions before we get going? Okay. So let me move this up and then share my screen. And of course it went on the wrong monitor. All right. Aha, all right. Sorry, it overrode my entire Zoom session, and I had to go re-find it. 'Cause, yeah. All right, is that working for folks? Okay. All right, and of course, the way that, oh wait, now I am able to see everyone's faces. All right, that's helpful. Yeah, Zoom makes it minorly difficult to see people's faces, but I think I've gotten that. All right, so, yes. All right, so I assume most of you have at least heard the term DNA before. And one of the first things that I want to do is just talk a little bit about what DNA is and why it's interesting. And so I figured I'd put up a relatively low resolution picture of what DNA looks like. But fundamentally, almost all information in known life on Earth is stored in DNA. And the way I say one of the reasons I say almost all is some simple organisms store information in RNA, right? And we're going to talk about what that is in a minute. But between DNA and RNA, this is the primary way that life on Earth stores information. And we can talk a little bit about what the differences are, etc. All right. And DNA, if we look at what it is, it's actually a sequence and an alphabet of size 4, all right? And it turns out there's 4 molecules that code for the bases, the alphabet. So there's adenine, there's thymine, there's guanine, and there's cytosine, right? And I'll expect Mike to jump in anytime I say things that are incorrect.
I'm in the process of typing a correction in the general notes.
Oh, okay, what is that?
Technically, the things that store information as RNA are not considered to be organisms.
Really? So simple viruses are not or phages.
Viruses are generally defined to not be organisms. Organisms are defined to be cellular.
Okay. Now, while Mike is almost certainly correct, I have to disagree with the biologists for classifying things that way.
I disagree with that classification as well, but I'm telling you what the classification currently stands as.
Sounds good. All right. So whereas in most of what we're used to in computers, everything is fundamentally stored in a base 2 encoding, DNA simply stores it in a base 4 encoding. All right. And if we look at a single DNA strand, we'll typically abbreviate those bases as A, C, G, and T, right? That's the easy way to view them, and they're bound together along a chain of sugars and phosphates, right, for a sequence. And it would look something like this. So where you have the backbone, and then you have all the bases attached to it, recording whatever information in order. And there is a directionality to this backbone, and you'll often hear reference to the 5' end and the 3' end, representing the 2 ends of a single strand of DNA. And I will let Mike explain, because I forget why they're called 5' and 3'.
So in organic chemistry, each individual atom has a defined number when you're looking at a sugar. The sugars that are involved in DNA have 6 carbons in them, and those carbons have a specified numbering of 1 through 6. The 5' end is the one where we have the thing that is attached to the 5 carbon at the end of the molecule. And the one that has the 3' label is the one where it's the number 3 carbon that's at the end of that molecule.
So fundamentally, the important thing is that this provides a unique beginning and end effectively to a strand of DNA, right? The 5' end and the 3' end. All right. So DNA is often described as double-stranded, and that's because these strands will wrap around each other, and you get something that looks like this. Whereas the two strands wrap around, the information in them matches up, and we're going to talk about in a second how it matches up. All right, and that is that everywhere there's an A on one strand, it will bind with a T on the other, for if it's a perfect binding. And everywhere there's a C on one strand, it will bind with a G on the other. So A's and T's match each other, and C's and G's match each other. And this is just a depiction of how these molecules line up and why A's match with T's and C's match with G's. All right. So what that means is when you have a double helix of DNA, the actual bases are different on one strand and the other strand. However, they match up perfectly, so the same information is effectively recorded in each strand. All right. Does this make sense? And when the DNA binds like this, it is extremely stable, right? It is very, when the DNA is in the double helix form, it is extremely difficult for a mutation to change any information on there. Right? And I like to equate a DNA sequence to a hard disk, right? Where a hard disk, you will have information on there that will last a long time. It might be slightly more difficult to access, but it is also good long-term storage. All right. All right, and when you actually look at the sequence, this is what I was describing, where the C's and G's match up and the A's and T's match up in order for it to all fit together. All right, so let's talk a little bit about DNA replication. Right, so how is DNA copied into a new DNA sequence, and particularly if it's all tied up in this double helix? And it turns out what happens is the double helix will actually unwind as it's being copied, and each strand has its complement strand built around it, right? And so as the whole thing unwinds and these new strands are created, you end up then with two copies of the entire double helix. Okay. So far, so good with all this? Okay. So replication begins when a snipping enzyme starts to cut the strands apart so they begin to unwind. And then lots of nucleic acids are available in the environment. And since the nucleic acids bind together in the way I was describing, that helps the new strand form. And I know there are lots of proteins involved in all of this to make sure it happens correctly. And even on top of that, there are all sorts of factors that I'm kind of glossing over, such as error correction. So error correction is a very important piece of all of this to make sure that during this process, changes might be extremely easy to occur while the copying is going on. Something, the wrong base can get in there pretty easily. And there's lots of mechanisms to go double check as things are moving to make sure everything matches correctly and mutation rates are lowered. And this tends to be one of the big differences between very simple organisms and much more complex organisms, is the quality of the error correction that is possible there, which is why to have very long sequences of DNA, you will often need to have better error correction mechanisms, or else you have a much higher error rate, and that is hand wavy because there are wide ranges of mutation rates that occur in different types of organisms. All right. So far, so you guys are so quiet. All right, I'm just gonna assume everyone's following along perfectly. Thanks for the thumbs up. All right. Okay, so how is information stored? So DNA is broken down into regions that are called genes. All right, and there's a lot more in there than just genes. We're going to focus on genes. Genes tend to be one of the primary foci, because genes are where proteins are encoded, and we're going to talk a lot more about that. There are lots of other regulatory regions and other things going on besides just genes in there. If we look at how genes are organized, very loosely speaking, genes begin with a promoter region, which includes an operator. And then there tend to be multiple genes encoded back to back after that that are co-regulated together, right?
Hold on a second. This is true in bacteria. This is not how things work in things that are more complicated than bacteria.
Okay. I will then finish explaining how bacteria work and shift over to you, okay?
Yep.
So the promoter indicates, all right, this is where we start for expressing genes, right? For converting genes to proteins. The operator is there effectively to handle Boolean logic, right? So there are things that can block genes from getting expressed, and there are things that can excite genes to be expressed at higher levels. And there can be all sorts of logic there where A is there, if nothing's there, it won't get expressed. If A is there, it gets expressed, except if B is also there, right? You could have some very complex Boolean logic in this operator region. But when the operator is satisfied, the promoter indicates the start, and the operator gets satisfied, and then these genes will be converted to proteins. And that is how it works in prokaryote. Mike.
So the main difference when we look away from bacteria is that bacterial genes are organized in what we refer to as operons. So as you can see on Charles's slide, we've got 3 genes in a row that are under control of the same promoter and operator. And I can get into the details of the molecular biology, but basically you make one piece of genetic information from that, and you use that as the template to make multiple different proteins. When you're dealing with eukaryotes, in general, each gene is independently regulated, and you don't have multiple genes in a row that are under control of one particular promoter.
Okay, so that being the main difference, that you wouldn't have a whole group of genes.
Yep. And then there are molecular differences that do not matter whatsoever for the purposes of what we are discussing today. But if anyone is interested, let me know and I will answer those.
Sounds good. Okay, so how are genes actually used? Well, so this is basically what I was saying. So the promoter region signifies the beginning of a group of genes in a prokaryote, of a beginning of a single gene, I guess, in a eukaryote. And I realize I'm using these terms prokaryote and eukaryote, right? Does everyone know what those terms mean? So I can pick someone at random and okay. So prokaryote and eukaryote have to do with the type of cell structure, right? Where prokaryotic cells are much simpler cells. They don't have a nucleus, and I think the membrane, I should let Mike explain the difference.
In general, prokaryotic cells have less in the way of internal structures. There are certain internal structures in eukaryotic cells. So eukaryotes are everything you can see with your naked eye, and a decent number of relatively small things too. Prokaryotes are bacteria. They are much more numerous than eukaryotes. They are complicated in other ways, but in terms of the cell structure, they're actually much simpler. Prokaryotes do not have a nucleus. They also don't have any of the internal structures of a cell that have membranes within them. All of the internal structures of more complicated cells that have membranes inside of them are derived from ancient other cells that got engulfed. And so you have mitochondria inside your cells that are really important for you being able to get energy. Those are derived from an ancestral bacteria. And plants have chloroplasts, which are really important for them doing photosynthesis. Those are derived from an ancient algae that got incorporated into the plant cell.
And at some point, it would be fun to chat about major evolutionary transitions and individuality and stuff like that. But,
and then we will loop Anya and Alex into the conversation as well.
Yep. But the whole idea of how individuals change what they are, and so, for example, how mitochondria, go from free living cells to part of another cell type, is a major transition in evolution, right? Because it changes what it means to be a living organism when those sorts of events occur. Okay, so fundamentally, though, bacteria are prokaryotic, right? Human cells are eukaryotic, right? And as Mike said, anything you can see with the naked eye is going, anything made of cells that you can see with the naked eye is going to be eukaryotic. An interesting side point also, you have more prokaryotic cells in your body than eukaryotic cells, all right? And the reason for that is the sheer number of bacteria that live in our bodies, right? It turns out, I don't know what the latest numbers are. I know at one point they were saying there's about 10 times as many prokaryotic cells as eukaryotic cells.
Current estimates are between 7 and 10x.
Yeah. So many times more prokaryotic cells. And how are we not just sludge if that is the case? Well, it turns out eukaryotic cells are much larger, about 100-fold larger than prokaryotic cells. And so even though we have more individual cells that are prokaryotic, most of our body mass is still human cells.
They're also not perfectly evenly distributed. Your concentration of bacterial cells are going to be on any surfaces that are exposed to the outside and in any cavities or passageways that have an outlet to the outside. So your bones and your muscle and whatnot are going to be all your cells, but things like the insides of your mouth and your nose and your digestive system and your reproductive system are going to have a lot of prokaryotic cells around.
Yeah, and at some point, maybe Anya would want to give a talk about that whole interaction, only because it's all incredibly fascinating how our bodies work with the bacteria inside of us in a very positive, mutualistic sort of way in most cases, and only when things go really wrong do we get sick from that. But that's Anya's main area.
If you completely got rid of your intestinal bacteria, you would have major vitamin deficiencies, because there are vitamins that we can't synthesize that we rely on our gut bacteria to provide for us.
Right. And many different diseases have to do with our microbiome going off in one way or another. Thanks, Anya. Okay, so there are all of these genes, and we talked a little bit about how what will trigger a gene getting expressed. But what does that actually mean? How is a gene actually used? And it turns out there are two main phases to this. There's transcription and there's translation that are going to occur. So first, genes are transcribed from DNA to a more usable RNA, and then they are translated from RNA, and this is particularly messenger RNA, into proteins. All right, and one way of one metaphor that works well for this is that moving from DNA to RNA is like loading something from a hard disk into a volatile memory, right? And so into main memory. Main memory is less stable for the long term than a hard disk is. However, it's much more accessible, much more usable. You can do something with it right now. Does that make sense? All right. Now, the translation stage is taking that RNA and turning it into a complex, three-dimensional structure that has all sorts of different structural and enzymatic and/or enzymatic properties to it. And that translation is more like what you do in a compiler, right? So a compiler, you take a program and convert it into something that's actually useful and functional, et cetera. And this is a loose analogy because there are even some circumstances where RNA will do things directly. There's lots more that we can talk about on that front, but as a loose analogy, I think it works fairly well.
I do need to jump in at this point. Slight shorthand in language that was just used has the possibility to confuse students in ways that we've actually documented fairly well. When we talk about you have DNA and then you turn it into RNA and you turn it into protein, you're not actually transforming the molecule. You use DNA as a template to build an RNA molecule. You use the RNA molecule as a template to build a protein. When you have made the RNA from the DNA is still there. When you've used the RNA to make a protein, the RNA is still there. It doesn't get deleted in the process.
True, I was worried that the metaphor would screw up, but fortunately the metaphor is still good. Like when you load something in from your hard disk, you don't automatically erase the hard disk. When you're using a source code in memory and compile it, you don't erase that original source code in order to create the new compiled form, the new executable. All right, so transcription. So transcription converts DNA into messenger RNA. It's often abbreviated mRNA. And RNA is similar to DNA, right? So it's almost identical in many ways, except that the sugar phosphate chain binding it together has an additional oxygen, right? So it's the chain is a little different. And instead of thymine, uracil is used instead. So that's a separate molecule that binds in a very similar way, but is used in RNA, whereas thymine is used in DNA. I think it's an interesting quirk of biology. I think that there is that difference. But as I was saying, RNA is less stable than DNA, but this is good because that means it'll go away on its own, right? Once mRNA is used, it breaks down fairly easily. All right, and so here is just an example of an RNA polymerase that slides along a template DNA strand and pairs up all the complementary bases to produce some messenger RNA, right? So it just matches up along the DNA. So the polymerase, as it moves along, separates out the double strand just enough for the mRNA to make a copy, and then so you have this mRNA strand that then gets produced. Right? Is that generally clear?
Can I jump in to ask something?
Absolutely.
How do you know which strand you should copy? Is there something in the DNA that tells you?
Okay, so you see how on this video there's like a little 5' there, right? Mm-hmm. So there's always a directionality. So you I guess you go from on the DNA itself, it's the 3' to 5' end, right, Mike?
Yes, the newly synthesized strand is synthesized from 5' to 3' on the new one. Charles, can you go back a couple of slides to where you had the schematic of an operon?
Maybe, there.
So here, that promoter region brings in the molecular machinery that allows for this polymerization to take place. And that has a direction that it is capable of moving on this backbone. So that promoter site is important for the cell to be able to signal, here is a gene, this is what I need to make a protein out of.
So that will indicate the direction it has to go in and all that.
Yeah.
So when the polymerase attaches, it will attach in the right spot and move in the right direction.
And the interesting thing is you have genes on both of the two strands. They're just facing different directions from each other.
Which is pretty awesome.
And in some organisms, DNA is so compacted that the same region has one gene on one strand in one direction and a different gene on the other strand in the other direction from the same nucleotide.
yeah, there's a lot that can happen in terms of overlapping genes and craziness on all of that. We'll talk about things like frame shifts and all in a little bit, but there's this idea that you can actually have genes that are overlap both in opposite directions and even in the same direction in some types of organisms, certainly in some types of viruses that are apparently not organisms, you know. All right. Well, okay, fine. But apparently all these biologists knew this. But... All right. Any questions before I move on? Any more questions? And thank you for the question. All y'all should ask questions. Okay, that's just starting and stopping. There we go. All right. All right, so translation, so that we just talked about transcription, right? We talked about transcription from the DNA to messenger RNA. Translation of a gene converts the genetic sequence to a protein, and this is done by a ribosome. So a ribosome is actually about 50 different proteins, I think. So here's a picture of a ribosome taking in.
It's also not primarily protein.
It's protein and other forms of RNA.
Yes.
But it takes in messenger RNA and produces a protein, and I'll have movies of that in a little bit. So it's about 50 different proteins bunched together. I just looked this up. The ribosome consists of about 50 different proteins that are all bunched up into two balls that clamp onto the messenger RNA and convert it to a protein. I thought it was about 50 different proteins and 3 strands of RNA that bind it together.
The RNA is actually the primary component of it.
Maybe 3 very long strands of RNA? I don't know, yeah.
The 16S and 23S and whatnot are pretty long sequences.
Okay. So the ribosome moves along the messenger RNA, and as it goes, it builds one amino acid at a time. And so remember how I was talking about how DNA is base 4, right? We're used to binary, which is base 2. DNA is base 4. Proteins in almost all life use base 20, right? So there are 20 different types of amino acids. So whereas DNA and RNA are built of nucleotides, proteins are built of amino acids. There are 20 distinct amino acids that are used in most life forms. There's technically others that are used in weird forms of life, but most life uses 20 distinct amino acids, and thus their proteins are in an alphabet of size 20. And a protein is really just a string of these amino acids, but those strings will fold upon themselves into some very complex structures and even link together with other proteins and complex structures to form the structures, right, of our bodies, right? So both the physical structure and enzymes are also just proteins that are catalytically active in some way. Right.
Interesting fact that I like to bring up at this point in the talk. So a given gene will have whatever its genetic sequence is, and I don't know if Charles is planning on talking about genes versus alleles or not.
I wasn't.
But we have a gene and it has some sort of sequence to it. If you take that same sequence of amino acids and this rate at which you create this protein out of those amino acids differs, you can end up having it fold into different forms that have different chemical activities. So the same sequence can have different folded shapes that do different things.
Right, and there are different chaperones that get it to fold in different ways.
Yep.
And there have any of you heard of mad cow disease? I'm sure most of you have. Mad cow disease, for those of you who don't know, it's referred to as a prion-based disease. And what a prion does is it misfolds one of your proteins to fold the same way as itself, right? So all that mad cow is your own proteins being folded the wrong way, causing other proteins of that type in your body to also get folded the wrong way. And I think this is just fascinating to me, because I mean, it's not like it has a prion-based disease has its own genetic sequence or anything. It's just using the sequences that are already in your body and making them go wrong. And I don't know. And the disturbing part about them is that these proteins can be very stable, and so even under high temperatures and various things like that, it is actually quite challenging to purge, prions. So, all right, but yeah, that's a tangent. So right now we're talking base 20, right? Is where we're moving, and where suddenly there's physical structure and catalytic structure in a very prominent way for these.
Hold on 1 second.
Mm-hmm.
From the audience, do you guys all know what we are talking about when we say catalytic?
Okay, there's at least a couple of people saying no.
So catalysts are things that speed up a reaction without being consumed or produced in the course of this. They make reactions run faster. The vast majority of reactions that take place inside a living organism are catalyzed by particular proteins called enzymes that speed up the rate of a reaction anywhere from about 100-fold to about 10 billion-fold what they would occur without a catalyst. These catalysts are utterly essential for the reactions taking place in a short enough time frame and at a high enough fidelity for them to actually be useful for your cells.
And in many cases, the reactions can go in two different directions, and it would primarily go in one direction, and a catalyst can change it to primarily go in the other direction.
That's not exactly true. Catalysts speed up the rate of a reaction in either direction. Your body forces reactions into one particular direction by moving the concentration of the reactants or the products.
Okay.
Catalysts speed up a reaction in both ways, but if the product of one reaction is being consumed rapidly by another reaction, it will drive the whole situation forward rather than allowing it to go back in the reverse direction.
Cool.
And technically, catalysts are involved in the reaction in that quite often they are consumed and produced at the same rate. So that if you actually look at the mechanism, they do become part of something, but then they come off again at the final product.
Oh yeah, well they will attach and then detach, yeah. So I don't know about the rest of you, but I feel like I'm learning a lot. All right. So to move on, wow, that's low resolution. All right, maybe I'll just move on to the next. Now, the idea here is that as a ribosome is producing a protein and moving along the messenger RNA, the protein is slowly getting built and slowly built, and it'll be folding as it's forming, right? And so these are little pictures of the ribosome moving along it. And then when the ribosome is done, it comes apart.
Yeah, I kind of have a question.
Yeah.
Can you go back to the last slide? So, with the ribosomes coming in, is there a certain amount of the like stools they have to go through because they're coming at like different places?
So what this diagram is representing is that, let's say you have a gene that you need a lot of a particular protein for. From that one stretch of DNA that you have in the cell that has that information, you can create multiple different mRNAs. And then each one of those mRNAs can even have multiple ribosomes moving along at the same time. Notice that those kind of coiled brownish yellow chains are different sizes. The ones that are longer are ones that have been on the mRNA for longer and therefore have created more of a protein coming out of them. This is part of what is referred to as signal amplification, that you can go from just that one DNA molecule to a bunch of RNAs, and each one of those RNAs can be used to make multiple copies of the protein, so you can get a lot of protein in a short amount of time when you activate this gene.
And regulation will control all of that. So if something is very upregulated, it might produce a ton of messenger RNA for a particular gene. If something is downregulated but still active, it might produce just one or whatever. All right, so transfer RNA. So we talked about messenger RNA. Another type of RNA that is interesting is called transfer RNA. And it folds into this interesting, is it referred to as a cloverleaf type shape?
Yep, this is a cloverleaf.
Which, of this form, where there's a, each one of these is associated with a particular amino acid, and a particular sequence that it matches on the RNA. So ribosomes use these transfer RNA to convert groups of 3 nucleotides into a single amino acid, right? That means as it marches along, each group of 3 nucleotides maps to a specific nucleic acid. A specific amino acid. All right, and so, and I think I give these numbers on the next slide, but so how many different groups of 3 can there be? So if a nucleotide is base 4 and you put 3 of them together, how many different sequences are we talking about there?
We have an answer in the chat.
I can't see the chat.
So Dylan points out that it's 64.
Okay, thanks. Yeah, while I'm presenting like this, I don't think I can access the chat very easily. Okay, so there are 4 nucleotides. So if there's 4 possibilities for the first position, 4 possibilities for the second position, 4 possibilities for the third position, that means you put 3 nucleotides together, there are 64 possibilities, right? And given that there are only 20 amino acids, this is clearly a mapping that can work. Right? There's lots of extra possibilities there. So, right, and so I say all this here. So there are 4 different nucleotides, and these are used in groups of 3. Each group of 3 is called a codon. And so when you're looking at this messenger RNA, it's a series of codon, 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 codon. Each codon maps to a unique amino acid, and so it determines the sequence of amino acids that are produced. So we already talked about this, how many codons are there? And so there are 64 possible codons. We will talk about what that mapping looks like in a little bit, but there are 64 possible codons. How does this work if there are only 20 possible amino acids? Clearly, there has to be some kind of redundancy, right? Where multiple codons might map to the same amino acid. All right, so what this whole process looks like, so you see how there are 3, the codons are 3 long, so any particular sequence of 3 nucleotides will map to a particular transfer RNA, which will produce a particular amino acid for the sequence. And again, there we go. And so here's a much lower resolution, but it's actually forming the chain there. So you can see how as the ribosome marches along, it's going to produce this whole chain, which is the protein. All right. This is me just grabbing things off the web that I was like, oh, look, that shows the point I want to make. All right, which brings us to the genetic code. Most of you have probably heard the term before, the genetic code. This is what on the right side here. This is what the code actually looks like. So when people say the genetic code, what they're referring to is this map of how the codons translate into the amino acids, right? And if we look at this map a little bit, one of the things that we'll see, so there's you look at what's in the first position, what's in the second position, what's in the third position. So if we have UGG, it's U, U, G, U for the first position, G for the second position, G for the third, and we have TRP, which is tryptophan. Okay, so that, and so we can look up what each of these code into. Here on the left side is a list of the 20 amino acids. Here's the 3-letter code for each of them that's used in the chart on the right. And for all of these, often when we're abbreviating a protein sequence, we will knock these down to just a single letter long so that we can clearly show what a protein sequence looks like easily. All right. Now, when looking at this code, it has some interesting properties, right? First of all, one of the things you might notice is that 3 of these codons code to something called stop, right? The stop codon indicates, hey, we're done with this gene, right? That's how it knows when it's done. When it hits any one of these 3 codons, it knows it should stop translating the gene. Does that make sense? And then there's the 61 other codons code to the 4, code to the 20 amino acid. Now,
interestingly, these stop codons, when Charles referred earlier to the fact that almost everything uses this set of 20 amino acids, the handful of organisms that use something other than those 20 encode that additional 1 or 2 from one of those stop codons. So there are organisms that one of those stop codons is in fact turned into a different amino acid that is available for their use.
So the genetic code is disturbingly stable across life. And there's a lot of question about why is it so stable? Why don't we just see the code rearranged more than we do? Right? There are minor little tweaks, but this is what it looks like. And I don't know, to me, that's crazy. There are some cool properties. So for example, if I said I'm going to change one of the codon positions, the first position, the second position, or the third position, I was going to randomize one of them. Which one would you want me to randomize if you wanted me to minimize the chance of changing what amino acid it coded for?
So I will monitor the chat if no one wants to speak up.
Could you repeat the question?
Sure. If I were going to randomize one of the bases, one of the codon positions, would changing that first position change the amino acid the most? Or be most likely to change the amino acid? Would changing the second position change it the most, or would changing the third position change it the most? Or change it the least? Either way, yeah, I guess. We could look at that either way. Let's first go for change it the least. Can anyone find a mutation that would not change it? Let's start with that.
So in general, we're getting mostly correct answers here, not all. But in general, people are saying that, yes, changing the third position is the least likely to change things.
Okay, so again, sorry, I still can't see the chat. All right, so if we look at all of this, let's just pick any one of the, I guess I shouldn't say any one of these, but let's pick, let's see, PRO, proline. So proline is I believe it's only these 4, yeah.
It is those 4.
So if we have a C in the first position and a C in the second position, does it matter what the third position is? Right? You could change the third position to anything and we'd still get proline. Does everyone agree? And you'll see anytime one of these boxes has all the same thing in it, like CG, anything is arginine, right? Now here's an interesting thing with arginine. If we had CGG, right? Clearly, changing that last G wouldn't matter. Would anything else not, are there any other mutations that wouldn't matter?
Changing the, oops.
Changing what?
That was Diane, so she decided to stop answering.
Okay.
I know the answer, but I would rather a participant answer.
Sure. Is there any other change you could make that does not involve that third position? If you started with CGG, so you had arginine, is there any other change you could have made that wasn't in that third position that would have kept it at arginine?
If you change the C to an A and have it be AGG.
Absolutely.
Yeah.
So
and this is why I had said that not all of the answers in the chat were correct. There are a lot of people who are saying that the first position is the least likely to change it. I'm sorry, the most likely to change it. It's actually the second position. There are no pairs of things where if you change the second position, it doesn't change it, with the exception of 2 of the stop codons both being the same, even if you change this 1A to a G. Every other time you change the second amino, the second position, it will change things. There are a handful of cases where changing the first one won't change it. So the arginines that we were talking about and the leucines, so the CU whatever also are the UU and either A or G. So there are a couple of times.
And glutamic acid, right? Oh no, I'm looking at
Nope, glutamine and glutamic acid are technically different things.
Yep, I was looking at it wrong. Okay, never mind. Yeah, so first position almost always changes it. Second position always changes it. Third position often does not change it. Okay. So it's an interesting coding. One of the things that is not as obvious in this chart is that if you look at the properties of these different amino acids, when you do change from one amino acid to another, it is disproportionately more likely to change to another amino acid with similar properties than it is to change to one with radically different properties. When you look at hydrophobic and hydrophilic, I forget all the different properties that they rate these on.
They're rated on size, how well they dissolve in water versus oil, whether or not they are charged, and whether or not they're polar, which has to do with their solubility, but is also slightly different.
Yeah. So often, even when the amino acid changes, the protein that it produces will be similar to, in terms of functionality, so that you can still have errors and the protein goes on and does the right thing. And so there have been lots of studies where they've tried randomly creating other genetic codes, and the genetic code that we actually have tends to be better than, by even simple metrics, tends to be better than I think all but 1 in a million randomly generated codes. And even that 1 in a million, when you start looking at it deeper, there tends to be some obvious reasons why the genetic code we have is better. Okay. So it is clearly a very good code, although there's lots of arguments for why it's not a perfect code, and that you could handcraft better codes. So why hasn't evolution jumped to the better codes? We don't know. I mean,
we have lots of theories, but we don't know for certain.
Yeah, there's lots of theory, but it's still an open question trying to understand that, which I think is amazing. But I remember when I was first learning about the genetic code, I had always assumed that like the actual sequence is what you talk about when you're talking about the genetic code, not that, oh, it's actually a code. It's a code in the sense of here's a set of symbols and how do they translate to this other set of symbols and so. All right. So, so there are actually only 61 codons that code for amino acids, as we talked about, hence only 61 unique tRNA structures. Right? The other 3 tRNA possibilities aren't built. And so the remaining 3, as we said, are stop codons, right, which indicate the protein's finished being constructed. And almost all known life forms use the exact same genetic code, and even the few where it differs, it's still mostly the same. There are just minor differences. as Mike was pointing out, that sometimes there's an additional amino acid that's used in the place of a stop codon or something small like that.
And sometimes there are very slight differences. So for instance, we talked that the mitochondria in your cells that are providing you a lot of energy are derived from an ancient bacteria. They have a slightly different codon table than the rest of your cell does. So there are a handful of small differences.
Which is crazy, right? Like within ourselves, there's that difference.
Yeah, and it's not just within your body, it's within a given cell. Portions of it do different things.
And so one of the real things with any time you're talking about biology, if anyone ever says biology always does X, they are almost certainly wrong, right? If ever anyone gives you an absolute about how biology works, because there's exceptions to everything, and usually they're really crazy, bizarre, interesting exceptions, but it's insane, the number of exceptions to every rule somewhere. And the whole nature of the evolutionary process, where things are, things that work better are what survive, and things that don't work don't survive, means that there are a lot of things that seem so haphazard but happen to work extremely well. All right. It's an animation. Oh, it is an animation. Okay. Right here. Yeah, this was another depiction of a protein being formed. Oops, and I once again can't escape. There we go. Okay, so proteins. As a protein is built, it folds into a structure, and there are many levels to the protein process. So there's the primary protein structure. So this is the sequence of amino acids. When you hear about secondary protein structure, this is looking at where a protein binds to itself, right? And so that sort of pattern. Tertiary protein structure really takes into account the full 3D aspect of how everything folds together. And quaternary protein structure starts saying, okay, when you have multiple of these, how do they fit together with each other? Right? Have any of you heard about the protein folding problem? Okay, I see some people.
I did, in fact, post the link to the relevant XKCD.
Oh, excellent. So what's funny is I normally know which XKCD you're referring to, and this time I don't immediately have it off the top of my head, but I'll go look at that in a little bit. So the protein folding problem is a problem that I think it hasn't even been proven NP complete, in that we don't know if we can do verifications in polynomial time. Anyway, it's a very hard problem, right? And what we would love to be able to do is take a protein sequence, and from that primary structure, right, the sequence, be able to predict what at least the secondary structure is going to be, and ideally the tertiary and quaternary structure, because if we could do that, suddenly we can do much better protein engineering. And as a research community, our ability to predict protein folding has improved a great deal, as long as it's similar to something we've seen before. We're still absolutely terrible about getting a novel sequence and having any idea of exactly how it's going to fold. And it turns out there's a lot of complicating factors, because it's not a matter of just taking the sequence, because everything that's going on as that sequence is being produced matters. Because I remember that one of the big things that improved protein folding was the realization that as a protein is coming out of the ribosome, it's immediately starting to fold. So it's not the same as just take the sequence and see how the whole thing folds, because it's going to fold to a minimal energy state or something along those lines. Because the start of it will already be folding by the time more of it is produced, that can radically change the ultimate folding structure that it ends up in.
So this ties into the point I had brought up earlier about the rate at which you are creating this protein can alter what the protein does. So one particular example that we use in a lot of our educational stuff is there is a protein that's involved in your cell being able to synthesize one of these amino acids, the amino acid tryptophan. If you have a lot of tryptophan, your cell can make this protein relatively easily because it has a lot of tryptophan inside the protein. And so if it makes it quickly, that causes the ribosome to just keep moving, and therefore everything folds as it's coming out with no problem. If you don't have a lot of tryptophan, the ribosome stalls. And so those helices and sheets start to interact with each other, and it causes the overall protein to assemble in a different shape. And so therefore, the shape that it makes when there's a lot of tryptophan available is not very active. And so you're not making a lot of tryptophan if you already have a lot. But the shape that it makes when it's not highly available is much more active, and so you make a lot more when you need it. I'll also point out this slide has the quaternary structure. Many proteins don't have a quaternary structure. Some of them do, and it's really important when they exist, but many of them do not have.
Yeah, that is a good point. I think my point here was just to say that there are many levels to consider when we're talking about protein structure, and some of you might have heard of folding at home. Folding at home is one of the classic now citizen science things where you get to try to predict how proteins will fold and all of that. Right, right. And so some proteins are just structural, while others will, enzymes will perform different sorts of functions, and so there are various types of proteins. Okay, and then gene regulation. And so most evolution over the past 300 million years has not created new genes. We will always say things like most, never all, right? For all of this. But most evolution has not created new genes, but instead focused on gene regulation, which is why when you look across different forms of life, organisms can look dramatically different and yet have such similar DNA because they share so many different proteins that are just regulated differently, causing the organisms to be built differently. But the underlying genes can be very, very similar. All right. So ensuring that genes turn on and turn off at proper times is not an easy thing. And so there's a whole big aspect to all of this about gene regulation, right? And how does all this work correctly? So Mike's example there was a great one showing how gene regulation works in one particular case where you have, if you have too much of a substance present, it will inhibit the need for an organism to generate more of that substance, right? Lactose in E. coli digestion of lactose is a neat example because there's a couple of aspects of that. Lactose is hard for E. coli to digest as compared to glucose. So if glucose is there, E. coli has no incentive to spend the energy to digest the lactose. So it should only do that if glucose is not there. But it should also only produce these proteins if lactose is present, right? It shouldn't try and digest lactose if there's no lactose. So it needs to simultaneously detect that glucose is not there and lactose is there. And there's all sorts of weird mechanisms like what Mike was describing that it can use in order to, by default, inhibit the generation of the lactose enzymes. But if lactose is present, it interferes with that inhibition, if I understand correctly, right? I know this is a classic example, Mike.
Mm-hmm. And so this has to do with those operator sites that Charles was talking about before. Operon is a classic example where you have something that inhibits, but that the thing that is causing that inhibition itself changes shape when it binds to some other molecules, and those things have to do with the concentrations of glucose and lactose in the cell. So the cell can essentially use that as a thermostat to figure out how much of the substance do I need to make to be beneficial under the current chemical conditions.
So from my perspective, these are these are the main things I wanted to try and cover, just to give an idea of what's really going on below the surface here. If I could figure out how to exit from all of this.
So, Charles, you said you were going to cover frame shifts, and you
Oh,
did
no, I
not
said
in fact
we
talk
can't
about
have-
different types of imputations.
Yeah. So sorry, I was going to mention that when we were talking about codons, and in particular, so we were talking about how a codon is groups of 3 nucleotides, right? That or matches groups of 3 nucleotides. Well, it turns out if you shift over by 1 nucleotide or 2 nucleotides, suddenly you're getting a mapping to a completely different set of proteins, right?
A different set of amino acids. It will still produce a protein, but it'll be a different protein, but it's a different set of amino acids.
Right, so you're mapping to a completely different set of amino acids because of that shift of 1 or 2. And so that's referred to as a frame shift. And sometimes you can have 2 different promoters that are on top of each other, but shifted a little bit. And so you can end up with the same region of a genome encoding for two different, or even more than 2 different proteins. Of course, this creates some real challenges for evolution. So one of the things that I really have not talked about is how all of this interacts with evolution, right? So evolution needs a source of variation, and there are many different types of variation that can occur in this genetic structure that we talked about, right? So the simplest one is a substitution mutation. When you have a genome, you have a nucleotide at one position, and it gets swapped out to a nucleotide, to a different nucleotide, right? And that may or may not change the underlying protein. May not the underlying amino acid. Changing out the underlying amino acid may or may not change the function of the resulting protein, right? But so substitution mutations can affect a gene, but the substitution mutations can also affect the gene regulation and change how a particular gene is regulated when it gets turned on and when it doesn't get turned on. Beyond just substitution mutations, you could also have insertion mutations and deletion mutations. That's when a base pair gets inserted, 1 or more base pairs get inserted, or 1 or more base pairs get removed. And then there's all sorts of more complicated sources of variation. One of them is sexual recombination, right? Where you have sequences that get mixed and matched. So sequences.
1 second, Charles.
Mm-hmm.
So with the insertions and deletions that you're just talking about, relevant to that frame shift discussion, certain types of insertions or deletions matter more than others. If you insert or delete a modulo 3 number of base pairs, it's going to have a much smaller effect on the protein, because that either just introduces a bunch of amino acids or removes a bunch of them, but it leaves the rest of the protein after that alone. If, on the other hand, you insert something that is off by 1 or 2 from being a multiple of 3, that will change everything from that point onward. Molly now also has a question in the chat asking about the biological mechanisms for different mutations. The vast majority of mutations that occur when the new DNA molecule for the new cell is being synthesized. Sometimes there is that proper base pairing, and that's going to be the most stable state, but sometimes the polymerase incorporates the wrong base pair. And there are mechanisms for the cell to try and detect that and repair it, but they're not perfect. So you're going to have some mistakes that occur there. There are also mistakes that occur at other places that are going to be relevant to what Charles is about to talk about. So I'm going to let him finish with what he was saying there. Then you can go back into these other sources.
Yeah, I was mostly just talking about how there are many different possible forms of variation that can occur, right? So
yep, and you started talking about recombination, and recombination is a way you can get other errors introduced too.
Absolutely. So recombination, you end up taking 2 different organisms' genomes and taking some of, copies of some of one with some of the other, and mixing them together. And of course, that can be very traumatic on the genomes if it's not done extremely carefully. And there's many ways that can go wrong that I think Mike was going to talk about. And there's all sorts of other types of variation, just to say very quickly, because sections of a genome can be duplicated. So duplications are one. They can be moved to a different region. They can be flipped around. So as you can imagine, there's all sorts of different variations. Sorry, Mike.
So in terms of the recombination, so something we haven't discussed at all today, but something to be aware of. Again, pretty much everything that's made of cells that you can see with the naked eye is going to have at least 2 copies of its genome. So you are what are referred to as diploids. So you have 2 copies of your genome, one that you got from mom, one that you got from dad. So you have, for instance, on each chromosome pair, you have one of those chromosomes that came from your mother and one that came from your father. And here we're talking genetic parents, not your actual parents. Turns out your adoptive parents did not give you their genes. They are still actually your parents. But from a genetic standpoint, we're talking about just the people who gave you your genes. The chromosome that I got from my mother is not either the chromosome she got from her mother or the chromosome she got from her father, but it is a mixture of those 2. When organisms that undergo sexual reproduction are producing the egg or the sperm cells, they don't sort out into just these are the genes from one parent or these are the genes from the other, but they make new chromosomes that are an amalgamation of them, broken and recombined at different places. Everything gets much more complicated when you start talking about diploids. Avida is haploid, and we stick with that for good reasons.
Yeah, so to bring some of this into the terms of Avida, in Avida it's haploid. We can still have recombination, but it's haploid recombination, which is a much simpler way of doing things, where we take chunks of one of the programs, chunks of another program, and just swap them around.
And then some of these other sources of potential causes of these mutations, duplications and deletions can both be caused by the fact that the machinery that is copying the DNA and making a new one, sometimes it falls off the DNA strand and has to be reattached. And if it reattaches at a place that it's already synthesized, it will duplicate that section of DNA. If it gets attached a little bit further on, because these molecules are flexible, it will have deleted that source of DNA in between. So that's one major mechanistic source of this. There are others that we can go into detail if you continue to be interested after today.
Yeah, there's so much that we could talk about in terms of any of this, and in particular in terms of the evolutionary process and how evolution works, because that's something that is going to strongly affect all of these, all of the different projects that I think almost anyone here is right. Probably not all of them, but almost all of the projects here. Since ultimately the types of software that we're building is focused on evolution. But what's important is that in the natural world, the underlying representations used are information-based representations where changes occur in them. And then through the evolutionary process, so there's this idea of natural selection, right? Where some of these changes will allow the mutant organism to replicate faster or better or survive longer, or somehow be able to produce more offspring than it would have otherwise produced. Those are the ones that we will see more of. If a mutation makes it such that an organism can no longer mutate, no longer reproduce, then we will not see it anymore. And so evolution really needs to be understood as just sort of this natural process. It's not about organisms wanting to reproduce or anything of that sort. It's about success in reproduction are the ones we still see. Lack of success in reproduction, we stop seeing these organisms after a certain number of generations. And that's going to be the same in biological organisms or in digital organisms, unless we do something really weird on the digital side.
It's also likewise important to remember that mutations are not inherently good or bad. There are lots of mutations that can occur that will have no effect whatsoever on the organism. There are many of them that will be deleterious because they break something, but there are also some that improve the function of the organism in some way. And whether or not a mutation is beneficial, harmful, or neutral depends heavily on the environment. So there are mutations that are beneficial in one environment, but are harmful in another. So one of the really common examples of this is that most of the mutations that confer resistance to antibiotics for bacteria are things that are costly if there isn't an antibiotic around. They make it so that the bacteria has a harder time reproducing, it takes more energy, more of its offspring are dead. But these are really beneficial in the presence of antibiotics because the option is either spend a lot of energy to reproduce or die. It's better to choose the former option. It's also important to realize that a whole lot of our knowledge of this is fairly recent. Now, what qualifies as recent in biology and what qualifies as recent in computer science are two different things. But some of this is still stuff that every year when I teach some of the more advanced genetics concepts, I spend a weekend going through the most recent year's literature to see what has changed since the last time I taught it, because I want to make sure I'm not telling something that's wrong.
So we have a little under 10 minutes left. We can answer a few more questions if people have other questions about anything.
Actually, a question about the gene mixing process that
Yep.
Mike talked about. I just posted that question in the bio channel as well, but since you have time to answer, I can just ask right now. I'm not a bio student, but common sense tells me that if genes are really mixed up randomly, that could be harmful for the organism. So
Okay, so slight additional terminology information that's necessary based on this. So a gene is this region in your genome that has genetic information. You can have multiple different versions of a gene. So these are referred to as alleles. So for instance, there is one allele that causes you to develop in such a way that the bottom of your earlobe is attached to the side of your head, and there's another a different version, a different allele that causes you to develop in such a way that the bottom of your earlobe is detached and dangling downward. This is a standard human trait. There's variation. I'm betting if we got high enough magnification of everyone's screens, we could see which version anyone had. These are two different alleles of the same gene. When we talk about this mixing of some of the genetic information from your mother and some of it from your father, there are breaks that occur on the DNA sequence, but they occur in the same place on both of the copy from the mother and the copy from the father and get reattached with fairly high fidelity. So that is, you might have genes A, B, C from mom, and then gene D, E, F from dad. And if they have the same alleles, you're not going to notice any difference. If they had different alleles, you get one or the other, but you're probably not going to get two copies of the D gene because things are broken in different places on the two DNA copies.
So if I'm understanding this correctly, genes that determine the same trait usually combine together.
So
the gene that determines your earlobe would not be combined with the gene that determines your hand.
Right. So within humans, for instance, all humans, to a first approximation, because all is a terrible word in biology, but to a first approximation, we have the same genes as each other. We just have different alleles. And those genes are in the same places on the genome. And so when you're doing this recombination, you read along the chromosome from mom for a while, and then you switch to the chromosome from dad in the same place. And so you will end up getting one or the other allele, but you're not likely to get both of them on the same chromosome. Now, the reason I say 2A first approximation is, as Charles brought up, one of the types of mutations is rearrangement. So you can move a gene from one area of your genome to another. And so that can cause these differences.
And
But
those
to a
types
first
of mutations are
approximation,
much
that's
more
not-
likely to be harmful. Just to...
Yep.
Got it. Thank you very much.
Yeah, and I mean, so what's interesting is the whole system by which sexual recombination works is super careful in the exact sorts of ways Mike was just describing. And so I mean, the system has so much to it in order to make sure that things don't screw up in so many different ways that that could easily be screwed up. And so one of the big questions in evolutionary biology is, how did sex first evolve, right? Because when you have recombination, you add on so many huge negatives, right? I mean, just the fact that you're taking DNA from another organism and mixing it with your own in order to produce an offspring seems like that's gonna be traumatic, right? It's a very challenging process to do well, and so it seems like an incredible increase in mutation rate. But there's all sorts of other factors that biologists are studying. I will say, even with just that one factor, we have trouble in these artificial systems showing sex evolve without giving it other sorts of pressures to evolve. But in nature, there's also this other factor referred to as the twofold cost of sex. So if you think about this from a female's perspective, right? If that female were asexual, she would be able to produce X number of offspring. By that same female being sexual, that many offsprings produced, but only half of her DNA goes into each of them. But the female's putting in just as many resources. And
because
so,
DNA is not the primary resource that they're provisioning to their offspring.
yes. And just because there's so many resources in creating a new organism. And so basically, if that female were able to produce twice as many offspring, or twice as much DNA in her offspring, this would be a compounding effect. And so switching from a sexual organism back to asexual should have your number of descendants going up exponentially every generation, right? Twice as much DNA that next generation, but then 4 times the generation after that, 8 times, right? Going up exponentially. And so understanding how sexual recombination could evolve to begin with is really challenging.
So to put this in slightly different words, because I saw a few people with somewhat puzzled expressions. If an organism is reproducing sexually, on average, a woman will need 2 children to reproduce her full genetic information, because each one of those children has half of hers and half of whatever male was involved. If she was able to reproduce asexually, she'd only need 1 offspring to have, on average, her full genetic information copied into the next generation. So you need twice as many sexually reproduced offspring to get the same amount of your genetic information into the next generation.
Anyway, so that's one whole branch of interesting questions and studies, but yeah, there are many others that are worth talking about. All right, so time is just about up. Any last minute questions?
Orchids?
So I just heard the word orchids. Do you want
Just
me
it-
explaining about like the bee orchids and whatnot?
I was thinking about, of all of the new orchids that are created by having duplicate sets of genes.
Okay, so things that humans have bred intentionally often have different numbers of copies of their genetic information. So many of our crop species, for instance, it's not just that they have 2 copies of their genetic information. Strawberries, for instance, have 8 copies of their genetic information. This is a good way to make cells bigger faster, which can be helpful when you're trying to select for larger cells in, say, the seeds or the fruit of something. It turns out that this also happens for breeding for ornamental purposes. We're not eating the orchids, but we are intentionally breeding them for certain characteristics, which can involve substantial genetic changes over very short periods of time.
Well, and orchids are unique in that they have a tendency to have not just duplicate repeating sections within a chromosome, but they will have double the number of chromosomes.
Well, I mean, that's what I was talking about with strawberries, for instance.
Okay, I didn't know strawberries did that. I misunderstood your explanation.
Yeah.
Because sometimes you just increase the number of copies of sections that code for the same protein, but you still have the same number of chromosomes. That can happen too.
Yep. And interestingly, most of the seedless varieties of fruits have an odd number of copies of their chromosomes. And that makes it so it's actually very difficult for them to produce seeds. And so that's how you end up with seedless ones, because you would need, if you had 3 copies of your chromosome, so you'd need 2 of them to go into one of the offspring cells and 1 into the other one. And that would be done independently for every one of your chromosomes. And that germ cell would have to match up with something that had exactly complementary numbers of all of them in order to get the right amount of genetic information as the offspring. That is very, very rare. And so things that have odd numbers of chromosomes tend to be sterile and unable to produce the seeds, which we have intentionally selected for in our seedless fruit.
Okay, well, we should wrap up. Time is up. Thank you all for joining us for this. And feel free to continue to ask questions on the Slack or however else you want to. And yeah, this is fun. Bye, everyone.
Thank you.
Bye. Thank you.
Thanks.
No problem.
Farewell.
Farewell. Bye.